Hello, today I wanted to talk about the GTase crest, the Christine rat sarcoma virus gene. So, crest is found on the cell membrane and it is anchored by a hypervariable region, which is a secondary structure that I will get into later. Um, but essentially, it converts GTP into GDP, and it essentially tells the cell to grow and obviously promote cell growth. And essentially, whenever GTP is hydrolyzed, it tells the active site to turn off. Essentially, that's our off switch in a normal functioning cell. However, that is not the case in a mutated cell where it continually is turned on, which leads to oncogenic signaling, which is cancer, which is detrimental to the organism because you have unchecked cell growth, which leads to tumors, whether that be malignant or benign. So, as a whole, this whole entire structure is going to be our tertiary structure. It's a 3D part of the model. And then for our secondary structure, it's going to be the actual chains that are combining it. You have your alpha helixes, which are I demonstrated in the white. You have your beta sheets, which are in yellow. Over here, I have the variable region that I mentioned earlier that is actually anchored to the membrane. And over here is going to be my, what's it called? Oh, I'm sorry. My switch 2, which is basically the active site or the active version of the crest. And then I have the switch 1, which is the inactive version. So whenever it's not actually promoting the signal. And over here, which is what we're going to focus on today, is going to be a P-loop. And essentially what the P-loop is, it's a glycine-rich loop that binds phosphate groups, which are these little things, with ATP or GTP, which is going to be important for our active site, which is what I mentioned earlier. And I forgot to mention this earlier, but our primary structure is going to be the amino acids, which is essentially just the building blocks of your protein. So over here in your P loop, like I mentioned earlier, it's going to be glycine rich, and glycine is non-polar, and it is a lot no, it's a lot smaller, which is good for the protein because it's a lot more flexible. However, over here where the orange is, it is replaced with a cysteine, which is a lot larger. It's polar, um, and it is a lot bigger than the original amino acids, which makes the GTP active site a lot longer, which eventually will lead to oncogenic signaling because it's taking up more space, which can lead it for it to be active even without even without something actively being binded to the active site. And some limitations that I faced is obviously this is a bunch of pipe cleaners. The entire structure is made out of amino acids. Like I stated earlier, it is the building block of the protein, and there's zero way to really display that. Another thing I wanted to point out is also that the beta sheets, which are the yellow ones I pointed out, are typically more flat, and the alpha sheets are more of a spiral, and they look the same. However, they are two separate or different structures, and the only way I could I guess conceptualize that is by making them two different colors. And also I feel it is quite, what's it called, immovable. um the protein itself isn't in a fixed state. Like I said, it has something that it has a signal that binds to it that whether it activates or inactivates it, and that isn't really possible with pipe cleaners. And that is all I wanted to mention today.